At the top of the hour. Hello and welcome everyone to session number 15 of our special series on COVID-19, a series that we have dedicated to diagnostics in the pandemic. We all know that laboratory testing is an integral part of the strategy for most countries in terms of preparedness, alertness, and response to the identification, management and care of cases for COVID-19. So as ASLM and our partners, in response to this, we have dedicated some sessions in this series to COVID-19 diagnostics. Today, we have the pleasure of hosting two of our esteemed manufacturers to share with us what they have, what we are using in terms of testing for SARS-CoV-2. We are going to learn the technical issues regarding these instrumentations, these assays. We would also want to mention that this platform is mainly for discussing technical issues, and we will try to desist from discussing issues that may be sounding like marketing. This is going to be a purely scientific platform. My name is Anafi Mataka. I'm from ASLM, where I function as a project lead for our United funded efforts in accelerating innovative diagnostics into national health programs, as well as a project manager for our laboratory systems community of practice funded by the Bill and Melinda Gates Foundation. A few logistics for the session. This will be a 1 hour long session, which will be made of two presentations from two manufacturers today, following which we shall go into a question and answer segment. Today we have Inqaba Biotec from South Africa, as well as KH Medical company from South Korea. To kickstart our presentation today, we shall start with Inqaba Biotec, and we have Dr. Aron Abera, who is the technical support manager at Inqaba Biotec Industries, and he has been there in that position since 2015. He also has their SNP genotyping and qPCR department, and he is originally from Eritrea and completed his undergraduate studies at the University of Asmara in Eritrea. In the year 2001, he was awarded a scholarship to pursue his postgraduate studies in South Africa, where he completed a master's degree in human genetics, and then a doctoral degree in medical biochemistry, and a 5-year postdoctoral research in the Department of Medical Microbiology at the esteemed University of Cape Town. Dr. Abera, over to you.
Thank you, Anafi, for introduction. Thank you for everyone for actually zooming in to hear regarding the talk that I'm going to give today. My name is Aron. So today what I'm going to talk about is I'll give a bit of introduction about our company Inqaba Biotec, and also what kind of solution is Inqaba Biotec offering for diagnostic of the SARS-CoV-2 in molecular ways. So to give you a brief introduction of the Inqaba Biotec company, we call it as an Africa genomics company, and our main office is in South Africa, Pretoria. The company inception the idea came in 2000, actually, and the idea came from our executive director, Dr. Oliver Preissig, on making of oligonucleotides. So that started in 2002, where our company Inqaba Biotec started producing oligos, primers production, and also start giving services for Sanger sequencing. So, and then we've been growing since then. We were the first company in 2007 to give next generation sequencing services using a platform from Roche. From then, our NGS services has also grown and currently we're using a Lunar system, and also a PacBio system that I'm going to explain later. In 2010, we opened our East Africa subsidiary that is based in Nairobi, Kenya. So it oversees all our East African countries. And also we opened recently an office in Tanzania as well. In 2014, we opened our West Africa subsidiary that is based in Ibadan, Nigeria. So that also serves our all West African clients. In 2016, we became also distributors of endorse. And again, 2017, we start distributing for PacBio, which is another next generation sequencing platform, which you can sequence long reads. That's generally about Inqaba. As I explain later, we're based in Pretoria. In the middle, that's our old offices. We call it the greenhouse. We're based here. And then 2 years ago, we opened a lab on the top left, a new lab space that all our services, including our Sanger, NGS services are done in this lab. We also have an oligosynthesis lab. It's using a machine called Mermaid, and we produce quality oligos. As Anavi explained in the introduction, I also run the single nucleotide polymorphism genotyping department. We use a mass array system for that. We also have an automation system from Eppendorf that we plan to use for extractions and other molecular related work as well. So this is a brief introduction of our main office in South Africa. As I explained earlier, we have an office in West Africa and also in East Africa. So we serve for the whole sub-Saharan continent, and we have a system inbuilt in terms of delivering reagents and consumables to a whole of Africa. So to get to the main point, I'm going to talk about our diagnostic tools for the current pandemic situation. As we all know, we've been hit by COVID-19 disease for the last couple of months, and then the pandemic has been increasing quite alarmingly. So a diagnosis tool that is accurate and also doesn't take a long time has been very important. So to just give you a background on the virus of COVID-19, it's called the Severe Acute Respiratory Syndrome Coronavirus 2. That's the SARS-CoV-2. It's a single-stranded RNA virus. It's a positive strand, and it's a cause of the COVID-19 disease. I'm not going to go in much detail about the structure of the virus, but I've put this picture from a Corman ETL publication because the regions that are highlighted here are the regions that are used on molecular diagnostic tools. So this is the virus in comparison to the SARS-CoV. That is the old virus, which is the one that's found in Wuhan. It's called the SARS-CoV-2 on top. And then you can see it has two open reading frames, open reading frame 1A and open reading frame 1AB. And the main parts that people use for diagnostic controls are, you have the RDRP, that's the RNA dependent RNA polymerase region. We have also the E region here, that's the envelope region here, and also what we call the nucleocaspid region here. So mainly primers and probes have been targeted on the N gene and the E gene, and also there are some that are using as well the S gene to detect the virus as well. So what is the approved workflow of SARS-CoV-2? So this is an RNA virus, which means, as we all know, RNA is not very stable. So the most important part is to collect the specimens in the right way. So, when I'm going to discuss about what kind of solution that we have that from Inqaba Biotec, from specimen collection to analysis and what we offer. So the integrity of the sample is very important upon collection, and it's also, as we know, the SARS-CoV-2 is very infectious, so it has to be a solution that can maybe inactivate the virus as well. Then after that, we have extraction of the RNA. So it could be used in a manual extraction method or in automated fashion. I'm going to discuss about what kind of solution we have here as well. After the RNA extraction, I'm going to talk about what kind of analysis can be done to detect the presence or the absence of the virus. So in terms of specimen collection, as mentioned earlier, the big challenge is safety regarding the specimen collection, because it has to be, it's a contagious virus, so there needs to be a safety regarding the specimen collection, and also the handling and transportation. I mentioned RNA is very unstable, so if it's not stored in the right way, it can lead to false negative results. So this is the challenge that is commonly we have on the specimen collection. So our solution on this is we are a distributor of a company called Zymo, and there is a DNA RNA shield collection devices that we offer from Zymo. This DNA RNA shield is well known to deactivate any infectious agents. It's been studied as well that it deactivates the SARS-CoV-2 as well, so to make it safe. In addition, it actually keeps the integrity of your RNA as well upon transportation. So we have different DNA RNA shield collection tubes for swab, and also we offer the DNA RNA shield as a solution as well. Upon collection, it can be added to the samples. So, and also our Zymo research have confirmed that the SARS-CoV-2 RNA is stable in DNA RNA shield for 28 days, which means even if you transport it at room temperature, it would still be, you'd be able to detect it. So it actually, you won't get a false negative results. And also the samples in DNA RNA shield can be shipped at ambient temperature, which actually minimizes the need for cold chain. So this is what we offer in terms of the specimen collection. The next one is after the specimen is collected, there's an RNA extraction step. The challenge with RNA extraction is it's a low throughput RNA extraction, and some of the RNA extraction kits are not optimized for biological fluids or clinical samples. This is very important. If you have a bad RNA, then you have a negative result, which a person could be SARS-CoV-2 positive, but can be reported as negative. So it's a very important that we have a good RNA extraction, and that RNA extraction kit is compatible with different sample types. So again, the solution that we have is from Zymo Research and also NAB. First, let me explain that what we have from Zymo Research. We offer our RNA extraction and purification kit, whether manually or that is actually a MAC-bid format for automated systems. I'm also showing a picture of our one of EpiMotion here that we offer from Eppendorf that can be used alongside our Zymo extraction kit to actually do automated extraction. So the Zymo research kits can be used for automated ones and manual RNA extraction. And also they're optimized for different biological fluids, whether they are from upper or lower respiratory specimens. And they've been also shown to be compatible with different transport medias, like universal transport media or viral transport media, could be saline or PBS and others. So the system is compatible with all transport media. And also we offer an extraction kit for New England Biolabs as well, that is based in US as well. It's called a Monarch Total RNA Mini Prep Kit. So these solutions that we have can give a good RNA, high quality RNA for a diagnosis. So this would be the second part of the diagnosis with the RNA extraction. So as I explained earlier, it could be a manual on the left, or this could be a MacBead approach with automated system, and we supply an automated system from Endorse as well. So after you have the RNA, you want to detect the virus, and the viruses are detected using specific primers and probes. And I think we also, as Inqaba Biotec, we synthesize primers and probes in our main office at Pretoria here. So these custom primers and probes are designed for different regions for SARS-CoV-2. So usually our clients will ask us to synthesize a specific primer and probes, and this can be done with speed, accuracy, and purity. And we have the capacity, the machine, as you see, is called the Mermaid machine, to produce high quality and high number of primers and probes. So these probes are compatible to different qPCR machine, and they use for multiplexing several targets of SARS-CoV-2. As I explained, it's needed at least maybe 2 or 3 targets to detect the virus. So we can actually produce different probes, and also to check any extraction control as a human internal control, like probes like FAM and HEX and others. So these are produced internally by demand from our customers. So the other part is the facilitating detection of SARS-CoV-2 with individual reagents. So this is, you have your RNA, good quality RNA that is extracted, and also your primers and probes, but you would need reagents to actually do the detection. So what are the challenges of that? After getting the RNA, there might be an RNA purification step needed, or in some cases, there might be crude samples that are extracted. So this adds to protocol time and cost per sample. So that's one of the challenges we have. So from this, we can offer from New England Biolab RNA detection reagents. So I'm gonna show you 3 solutions that we have. The first one is a Lunar Universal Probe one-step RT-qPCR kit. This is a hydrolysis probe-based detection of RNA. So you can use it in combination with our primers and probes to do multiplex reaction. And you can use the RNA directly to it because it's a one-step RT-qPCR kit. And this takes about less than 2 hours to do the assay, and it's been validated by using different targets of the SARS-CoV-2, and also by multiplexing different probes as well, so it works very well. The other kit that we have is the Lunar Script RT Supermix kit. This is when you plan to do a 2-step RT-qPCR workflows. So you have the RNA and you want to synthesize cDNA from the virus, and then do another step of detection, or could be another maybe next generation sequencing as well. So this kit can be used to synthesize cDNA of the virus, and it can synthesize up to 3 kb. So you can synthesize the full viral gene. The third step, the third solution that we have is the Womstat colorimetric lamp master mix. This is quite important because it can be used on crude samples. This is, you don't have to have a very pure RNA, so crude samples can be used, and also it's very rapid. So it's a one solution mix that can do reverse transcription and also isothermal amplification. So this can be done in one temperature, which actually then if you have even a water bath, you can use this kind of solution. And also the kit that we have can be detected with simple visible detection as well. So it's a change of color. If you have the solution is pink solution. If you have SARS-CoV-2, then the color changes to yellow. So it's quite a simple visible detection, and it can be used in fields as well. So all the solutions are from New England Biolabs. What about in terms of sequencing the COVID-19? This is mainly for research based, not the Illumina and next generation sequencing are not used for diagnostic reasons yet. One of them is Illumina sequencing. So we can actually give the service of Illumina sequencing of the virus and also offer reagents that are needed to do the sequencing. So there are already published primers called the Arctic SARS-CoV-2 primers. They are about 400 base pair in size range. And then you can actually do the PCR, pull them, and then sequence them on Illumina. So these are the reagents that we offer to doing that kind of sequencing. But if clients are interested also in sequencing, we can also give the services here at Inqaba Biotec. The next one is a long read. We call it a PacBio Hi-Fi sequencing. This is to sequence the full region of the virus. So this is a long Amplicon virus sequencing. We are planning to give services as well for clients that are interested on sequencing the full genome of the virus. This is a nanopore sequencing, another quite commonly used technique. This is Amplicon sequencing protocol for mini-iron for SARS-CoV-2, and we offer the below reagents here to do that analysis. So you can actually contact us if you're interested in any one of these reagents or services, then we can give you more information on that. So this is the general part that I wanted to discuss. So bit by bit, we offer kits for Rna extraction and also primers and probes for detection, also reagents to do the diagnosis test. But today I'm going to focus on one of the kits that we supply from a company called Seegene. This is an all-plex SARS-CoV-2 assay. It's actually from a company called Seegene from South Korea. It's a company with wide range of solution applicable in diverse markets, from clinical diagnostics to basic science research. Most of these kits are for IVD, for diagnostic researchers. So this kit can be used as well with auto extraction system, Seegene Nimbus system here that you can see on this side, and they're designed to use real-time PCR machines. So we have a CFX96 and an AB7500 PCR machine that can be used for the all-plex SARS-CoV-2. So this you need a specific software to do the analysis as well, that I'm going to go in detail. So this is a SARS-CoV-2 assay. This is an update of a previous kit. Seegene first produced a kit called nCoV-2 that was out sometime in January. It had 3 SARS-CoV-2 targets and an internal control. But recently, they've actually released an updated version of that actually targets 4 regions. The testing principle is real-time PCR, so you need RNA to do the analysis. It's a multiplex real-time PCR assay to detect 4 targets: the RNA-dependent RNA polymerase, RDRP, the S and the N gene, specific for SARS-CoV-2, and then you have the E gene that is for SARS-CoV-2, including the SARS-CoV-2. So by using actually 4 different targets, you actually make the assay very sensitive. In addition, an exogenous gene is also used as an internal control to monitor the whole process, from sample collection. Is the sample collected properly? Is the RNA extracted well? And also, how's the analysis? So if your internal control shows a negative result, then the result is invalid. So it has to be either repeated or a fresh sample needs to be collected. And this is just a picture showing which region that are targeted on this kit. The RDRP, the S, the E, and the N gene are targeted. What kind of sample types are used for the C gene? So you can use nasopharyngeal or throat swab, or a nasopharyngeal aspirate, or a bronchoalveolar lavage, and a sputum sample. These sample types have been validated to be used by this on-flex SARS-CoV-2 assay. What about the sensitivity of the assay? It can actually detect as low as 50 copies of the RNA virus for all the targets. So this is an improvement from the previous kit that they have, the nCoV-2, which was about 100 copies for target. So this is quite important and interesting. What about the cutoff values? As you know, qPCR analysis uses a number of cycles that it will take to detect the virus. So anything that is below 40 can be considered as positive. So if your results show results of a CT value of 40 or above, then the assay is invalid. The same applies for the internal control here. So what kind of supplies and accessories are needed to work with this all-plex SARS-CoV-2 assay? Obviously, you need the RNA stabilization media, the one that I explained. A shield can be used, and then Rna extraction kit manual. If you're using the Nimbus automated extraction kit, it can be used alongside the C gene kit as well. And obviously your laboratory consumables from gloves, pipettes, plates, and tips that are needed for. So all we can supply from our side as well. So this is what the kit looks like, the SARS-CoV-2 assay kit. So it contains an oligo mix of all of them, the 4 target gene, and a premix that would contain the master mix, the NTPs and others, and also the premix contain a UDG as well. A UDG is very important when you're doing diagnosis tests and also when you're processing a lot of samples so that you don't have a carryover contamination. So that's one of the advantages as well, that you have a UDG enzyme in the premix. So the kit comes as well with a control for the SARS-2, a positive control, and also an internal control that can be added to the samples to check a whole process from sample extraction to the detection. And it also contains an RNase-free water. So the kit comes as 50 reaction kit and 100 kit reaction, and should be stored in -80. And usually you shouldn't use it more than 8 freeze and thaw cycle, so that you can get optimal results. So how do you interpret the results? I explained earlier, the most important control that we have is the internal control. This controls the whole process. So if the internal control that works in the hex channel comes as negative, then the assay is invalid. It shows that there was probably samples was not collected properly, or the RNA extraction step didn't work, or the qPCR, the analysis didn't work. So this is one. Then you have the other targets here. You have your E gene, so the RDRP gene and the S gene are in one channel, and then you have the N gene. So if you have positive for all the 3 of them, you are the SARS-CoV-2 positive. If you have positive for E gene and N gene, that's also the positive. And also for the 2 targets, for the RDRP S gene or the N gene, that's also considered positive. For the E gene, RDRP and S gene, that's considered positive. So if one of them here is positive, the middle one, the RDRP gene and the S gene, is also considered positive. And also if the N gene is positive, then the sample is considered positive. If the E gene is the only one that is detected and it's positive, this assay needs to be repeated just to confirm, because the E gene actually can it can pick up other SARS viruses as well. So this needs to be repeated. So if you have positive for the internal control and negative for all the targets, then you have a negative. And then I explain later if if the internal control is negative, then the assay is invalid. And this is how you interpret the result. The assay, yes.
I just wanted to remind you, I think, to be mindful of time to allow the other presentation, we need to wrap up. Yeah,
this is my last slide. So this is my last slide. So the software the kit comes with the software as well with the analysis, as you can see, which ones are positive and which ones are negative, and then we'll do an auto interpretation to just say if you are a SARS-CoV-2 positive or negative. And this shows the different actually amplifications of each target. And this is what the interpretation looks like. And yeah, that's all I have. But if you have any questions, I'm willing to answer.
Thank you very much, Dr. Aron Abera, for the wonderful presentation. We want to quickly jump into the next presentation, so we shall take questions at the end of the presentation, which is coming from KH Medical. And to do that, we have Dr. Muhumuza, Julius, who is the technical advisor for KH Medical. He's currently working at KH Medical and is involved in the COVID-19 detection kit development and continues to provide technical advisory services for COVID-19 tests to other organizations and teams as well. He will be supported by his colleagues, Mr. Adam Hong, who is the CEO of KH Medical, as well as Dr. Rugola Mtandu, an epidemiologist and representative of KH Medical as well. Over to you, Dr. Julius.
KH Medical. We are a group of volunteers, mainly scientists from Europe and Africa, who are volunteering to provide resources in terms of knowledge for willing manufacturers to produce these kits at a more cost-effective manner for mainly the Sub-Saharan Africa and Asia and South American
Dr. Julius, the slides are not yet on, just to let you know.
Hello?
Here your slides.
Hello?
Yes, we can hear you. Yes, we can hear you. I would like to ask you to share the slides. We cannot see the slides.
Oh, okay. Just a moment. Apologies for that.
Thank you. Whilst he's sharing the slides, I would like to let everyone know that we'll be sharing the code for those that need certificates immediately after this presentation.
Are you able to see the slides now?
Yes, thank you. Go ahead.
Good. So I was quickly correcting the information shared that we are a group of scientists who are offering services, voluntary services to willing manufacturers to produce these kits in the most cost-effective way. So most of my colleagues are not employees of KH. They are employees of KH Medical who can talk about the company at the end. I'll concentrate on the science of this kit that we've designed. I will let James or Crystal, who's from KH directly, to have 1 minute to talk about KH, the company, and then I will do the introduction of the kit.
All
Crystal?
right. Thank you. Your voice seemed to be shaking a bit. Maybe whilst Priska is coming in, you might just check maybe the mic if it's fine. And a kind reminder to try and do within 20 minutes.
Sure.
Please go ahead.
Crystal, are you on? Okay, it seems that Crystal is not online or is not able to hear us. So I can go ahead with the introduction of the kit. And I'm right now displaying the agenda. I hope everybody is seeing what is on the screen. Please confirm and
Yes.
quickly go through what we have to discuss. As my colleague from Inqaba company has described, I will not go through all this detail. We know that the SARS-CoV-2 is an RNA positive sense single-strand RNA virus. It's quite large, almost 30,000 base pairs, translates into about 160 nanometers in diameter. And it has 10, to be very specific, 9 functional genes that have been isolated and identified. And for the Rna COVID-19 detection kit, which we designed, we are targeting the S gene, which is the spike, the gene that codes for the spike protein on the surface. And we are also using the RNA-dependent RNA polymerase gene, which is in the open reading frame A1. We used the special software to do comparison with other SARS viruses, other coronaviruses for homology, and selected specific regions that have less than 75% homology to design our primers. The kit is very simple in terms of applying it. Initially, we had not manufactured the extraction kit, but now we have a complete solution which gives you the Rna prep or extraction, RNA extraction kit, and then the amplification and detection kit. The slide shows you a very simple procedure. We have an automated, a fully automated desktop or benchtop platform we call the Rna prep. It is battery operated, very miniature, and that is what we use or recommend for you to do the RNA extraction using the prep kit that is provided by Rna, okay, KH Medical. And after you've extracted your good quality RNA, you use our kit, which basically has the master mix, the prime and probe mix, and you combine with the extracted RNA, and you can use it on to do RT-PCR using the radicycler. There are 3 radicyclers produced by KH. We have the mini cycler, which is able to do 8 tests in 80 minutes, or the 16 well, and the 96 well, which is a high throughput machine. You can also be able to do your RT-PCR using other cyclers from Biorad, the FX96, which is very common, the ABI 7500, the Rotagen from Quagen, and the light cycler from Roche. All these are compatible and have been verified and validated to use the Rna COVID-19 detection kit. So what is provided in the kit? The kit is intended for quantitative detection of COVID-19. The recommended specimen is a nasopharyngeal swab. You can use sputum, also bronchial lavage if you can get that. We target the RDPR and S gene, as I said, and the product contents include the 3X RT master mix. It comes with a yellow top. We have the COVID-19 prime and probe in the brown tube, and then we have the positive control and RNase free water. That's the package. The limit of detection is actually extremely low, 0.66 per microliter, copies per microliter, and the recommended storage is minus 25, minus 15 to minus 25 degrees centigrade. This is how it comes. This is how the kit comes. And you can see it is color coded, you know which one to use. And it is packed in the 50 and 100 tests packet. These are the thermocyclers or the RT-PCR machines that are compatible with this kit. We recommend, of course, the radicyclers, the Mini for 8 wells, the 16 well, and 96 well. The Mini and the 16 well are benchtop. They are very easy to deploy in the field, and they are very robust, and you don't need a high level by safety, given that, as I will talk later, in the extraction kit, we provide also a deactivating transport media for the sample. And these are the other PCR, real-time PCR cyclers that you can use from other companies. So in case you do not have what is provided by this company, you already have one of these. All you need is the kit, and you can move, be able to do your tests. And this describes, for any laboratory, and I think this is very simple, this describes the volumes you need to use. This is a very simple procedure. Everything is pre-made. All you need is take, from our kit, 10 mils of 3XRT master mix, 5 microliters, sorry, of the prime and probe, add 15 microliters of your good grade extracted RNA, and then you are able to run your test. This is the recommended amplification protocol, and we use FAM, VIC, and ROX. ROX for the internal control, and the S gene, we have embedded the FAM for detection, and RP, we have the VIC. These are fluorescent dyes for detection. This shows you how to prepare the master mix, but basically what I just described. So you can make your aliquots early enough, and because as you run the prep machine, you'll be preparing to do your RT-PCR. So in order to save time, depending on the volume of the machine you're using and the number of samples you're getting, this helps you to do your aliquots early enough so that you can move faster when you are working in the lab. This is how the machine will display the results. And later I will explain to you the cutoff limits or the CT values that we consider for positive and negative. For the S gene, which is embedded with the FAM and the fluorescent dye FAM, the CT value is between 20 and 30. This is what we're saying all the time, but just around, 21, 22. For the internal control, we usually say it around the same, 20, 24, 25, and also for the RDPR gene. So our threshold is set at 20. So any delay after 20, we'll consider that as invalid. And this is how we interpret the results, very similar to what my colleague described. And we have a cutoff value for undetected as 40 as well. So a CT value of greater or equal to 40 is what we consider as a negative. And this is how we interpret our results. It's very similar to what my colleague described. We are using 2 genes, remember, so for us it is either of the 2 being positive, then we'll take that as a positive result. If all of them are negative, including our internal control, then that's an invalid. If the CT values are above 40 for all, then it is invalid. If the CT values are above 40 for the internal control and one of the genes is negative, then that is invalid as well. So these are very simple chart, but fortunately, our cyclers are able to give you a direct result without going into the interpretation. But you can dig up this, in software, and it will be able to show you exactly how it showed up or how it came up with a positive or negative or invalid result. I want to talk also about the limit of detection. This kit has a very low limit of detection, very low limit of detection at 0.66 copies per microliter. And this has been independently evaluated by FIND and has also been compared with the available published WHO primers and probe. And it's been found to perform extremely well. I also wanted to share with you the independent evaluation carried out by FIND in February, when they put out a call to manufacturers of these kits to share their information and the kits for evaluation. The Rna COVID-19 detection kit was also shared, and the results are available on the FIND website. And this kit came out with 100% sensitivity, 100% specificity, and there are no cross reactions with other pathogens, 53 of them that were compared, and also all this COVID, all the coronaviruses that were tested with it. These are the results provided by FIND on their website for the independent evaluation. You can go and find this information, and the slides I think will be shared by ASLM after that. We have gone further to seek certification for this kit. There is CE certification already, ISO, of course, 13485 for the manufacturer. This comes from the manufacturer. They are certified. ANVISA, as of 1 week ago, it's registered with the Kenya PPB. It's pending FDA approval. It is being in process. WHO, EUL is also pending, and also in Tanzania we've submitted, and in South Africa we've submitted for registration as well. Now, about the prep kit. This is another addition to this kit, because RNA preparation is extremely important if you need to do your tests. And we have also worked on providing a kit which you can use for manual extraction, but you can also use it for the automated Rna Prep Plus machine that is also available. So we recommend that we use nasopharyngeal swabs. It can also use stool or sputum. And the method applied is the silica spin column based extraction. It's quite quick. You are able to get your prep sample within 25 minutes. The Rna Prep is able to do, to give you 8 samples in those 25 minutes. This is how the kit looks like, the prep kit. So in addition to this, we have added now a liquid amines based sample collection kit. And this should be able to stabilize the sample for about 20 days under room temperature for transport and storage purposes. But in the absence, you can use saline for transport. In that case, it won't stay for much longer. You will need only 6 hours to do the extraction. If not, then you keep it at 2 to 8 for at least 24, not more than 24 hours. These are the contents of the extraction kit. And it is also provided together with the detection kit. So it comes as a combination. The performance of this kit has been independently evaluated as well by CDC. And it was compared with the QueerGen extraction kit and found to be at par in terms of performance. The results for that evaluation are also available in this presentation, and available also on other websites. And this is the performance of the extraction kit, which also shows very good performance in terms of the limit of the copies in the sample. This is the automated Rna Prep Plus, which we recommend for automated nucleic acid extraction, RNA and DNA, but in this case we are talking about COVID, so it's RNA. we know that it's a very highly infectious virus. So if you use the transport media that is able to deactivate, then with you do not need a biosafety level 2. With proper PPE, you should be able to do extraction in automated extractor and use our benchtop solutions to run your test even in the field. This extractor is able to give you results, as I said, within 15 minutes, can take 25 minutes if you have another step, but basically 15 minutes. It's very lightweight, 18 kilograms. It is battery run, which is a rechargeable battery, which is a very good advantage for deployment in the field. And we can use stool dried blood spots for those who want to use it for other methods. But for COVID SARS-2, we use nasopharyngeal secretions. There are other products that the company is able to make, and I would let my colleagues from KH talk about this specifically because my portfolio really, settles on COVID. I let my colleague, Dr. Rugola, talk about this. Thank you very much.
Thank you very much, Dr. Julius, for the wonderful presentation. We are about to get into the questions. I see Dr. Rugola is there. You wanted to say something? Okay. Unmuted him. You should be able to speak now. All right. Whilst he is preparing, I think we need to get into...
Yes, Sanafi, thank you very much.
It looks like, can you hear us?
Hello?
Yes, go ahead. Hello?
Hello, Anafi? Yes, I can hear you.
Go ahead.
Sorry, sorry, my connection is kind of not stable, but I hope you can hear me. Yes, good afternoon, everybody, and good morning, everybody.
Yes, go ahead.
Go ahead.
Yes, yeah, so I'll try to take from what Julius has presented, but I missed some of the presentation. But what I want to say is a little bit on the tests, on the certification of our test, if Julius hasn't presented that. But just to say a few words, the test, the Rna COVID-19 detection kit has received a CE certification, and it has also got ISO certification, and recently it has just got the Pharmacy and Poisons Board certificate from Kenya, and the TMDA, that is for the case of Tanzania, application is pending approval, same as for the FDA EUA and WHO EUL. And we've also got ANVISA certification, that is for Brazil and Latin America. Just to jump into some of the questions that I saw them coming from the chat box on the logistics issues. Yes, these kits are ready to be deployed to countries in Africa and Latin America. And we have made arrangements with courier services, courier companies like DHL. So the kits are ready to be employed as long as everything is, all the administrative issues are taken care of. The kits are ready to be deployed. So that is much from my side. Maybe my colleagues, other colleagues from KH, Crystal, they can also present a bit on what is KH, where it started, and what other products are in the portfolio. So Anafi, if you can please unmute Crystal. I think she has something to present to us. Thank you.
All right, our time is running out a bit, so I think I would like to take a few questions a bit, and then maybe she can answer one or two. You provide me with the name. Let me take a few questions. We'll add a few more minutes than the usual, given the time that is gone. Dr. Aron, this question comes from Maubi Lirotodi from Lesotho. And he says, with this manual kits and setup, what laboratory level, 1, 2, 3, would be best for extraction and detection? What are the recommended BSc levels? I think that's what he wanted to say. You're on mute, Dr. Aron. Aron, you're on mute.
Okay. Okay. So the most important thing that I want to focus is the only one of the reagents that I talked about, DNA RNA shield, is the one that we know, or I can confidently say it has been shown to deactivate all pathogens, including the SARS-CoV-2. So we don't know how the specimen usually are collected or not. So it's actually is important to work in a facility that has the same standard as like TB labs or the BSL labs with the right protection, because we actually don't know how well the samples are actually deactivated in collection media. So it's important to work on this BSL-3, high BSL-3 labs.
Thank you very much. I'll take a second one from Kenneth, which might be applicable to both. For PCR kits, Kenneth says, for PCR kits that, this is quite a common question that comes, so it's important we take it. For PCR kits that mention that they are optimized for specific extractions and PCR platform, does that mean they cannot work on any other system? Should you rather choose kits that do not mention any special platform?
Okay, some of them, they say that they are actually work with a specific platform because they also are linked with like interpretations of the results automatically. So for example, our one-step reagents from New England Biolab should work with all instruments. And even the Seagen kit that I gave a talk on would work in any qPCR instrument, but obviously the programs that would give you automatic results are the ones that are validated with the kit. So you can, depending on what you need, you can use both. Like sometimes the kits that are diagnostics kits might be expensive, so you might be able to use the PCR kits without primaries and probes separately. So yeah, I can give more information if Kenneth can email me, then I can recommend what's the best kits to use.
Thank you, Aron. I know Dr. Julius mentioned a number of instruments, and then you made a recommendation for the RADI kit. So that also, would you want to just quickly maybe cement that point?
Yeah. Oh.
Yes, thank you, Anafi. As I said, you know, all that range of instruments that I listed can use the RADI kit. We've used it much more on other, on our RADI platforms, but we know laboratories which have these other equipment and are using the kit. And we've also done the verification using these other platforms, and it works very well.
Okay, thank you very much. I know we are almost on the dot, and I'll take maybe one or two more questions. What we shall do is to take all these questions to Aron and Julius for them to provide maybe specific answers in writing, and then we can quickly share. We will also share with you frequently asked questions that they have already prepared with regards to the products that they presented on today, in order to make as much, I think, information available as possible I would like at this moment to also share the code for those that would want to go and download the certificate of attendance on our ASLM website. You can see it on the screen right now. We have been having quite a number of questions regarding how the process goes. We appreciate this is relatively new and there are a few challenges, but our team, IT team and communications team have tried to break this down and put it on the website. But feel free to keep on checking with us until this is perfectly fine. For those that who may have missed, you can still go to the ASLM Academy and rewatch the session and follow the instructions that are there. I'd like to ask Aron just in 30 seconds to give a bit of 30 seconds to give some closing remarks.
Okay, I'll start first. Thank you for everybody for joining this session, and thank you, Anafi, for organizing this talk. I'm available on email, so if you share the email to everybody, so we can answer any questions that you have, starting from what is the most compatible, what kits do we recommend in terms of cost, prices, and any questions that you have. And also if they want to see the presentation, we can share the presentations as well. So from my side, thank you, and that's all I have to say.
Thank you once again. And from KH Medical, any one of you colleagues, Dr. Rugola or Julius, make some 30 seconds closing remarks.
Thank you very much for the opportunity to share this information, and you can please contact Dr. Rugola when it comes, or any other members of KH when it comes to talking about the prices and what is available, how quickly it can be made available to you. From the technical side, this is a very good kit, which we think will help a lot in sub-Saharan Africa. KH, I think, has very good packages as well for availing all this equipment together with the kits at a very cost-effective and timely manner. So thank you. You can send all the questions to Dr. Rugola.
Thank you very much, Julius. And what's left for me is to thank the presenters for a wonderful job, and also thank all the participants who came in their numbers. We hope this has been beneficial, and we shall continue to bring more and more of industry players, more and more of the technical content on COVID-19, as well as testing behind this pandemic. Till we meet again, goodbye, and good morning, good afternoon, and good evening.